Hi, everyone. Welcome to Water at Wayne. We are going to get started in a couple minutes. I'm going to give people a couple minutes to join, and then we'll get started. Hi, everyone. Welcome to Water at Wayne, our first seminar of the winter semester. Before we get started, I'm going to just go over a couple Zoom tips and housekeeping items. I'm going to ask that all participants mute themselves throughout the event so we don't have any random noises, so we can focus on our speaker talking. I'm going to ask that everyone keep their videos off throughout the event. And then anyone who has questions throughout the event, you can just drop them into the chat box and we'll answer them at the end of the event during the Q&A. I'm really excited to introduce Justin. Justin is the research coordinator at Ohio State University's Field Station Stone Laboratory, which is on Gibraltar and South Bass Islands in Lake Erie. The research in his lab focuses on cyanobacterial harmful algal blooms in Lake Erie and the environmental factors that promote bloom growth and toxicity. Also, he facilitates visiting scientists who visit and conduct research in their labs, use their research vessels, and deploy long-term monitoring equipment on their grounds. Additionally, he coordinates Stone Lab's undergraduate research program. Finally, he is also a certified merchant mariner credential by the United States Coast Guard. So thank you, Justin, and we're really excited to have you.
All right, everyone, thanks for inviting me. I'm going to go ahead and get started. I'm going to talk about a project that we've been working on for the past couple of years. I also work with Judy Westrick at Wayne State, and some of her data that she's helped us gather for this project is in my presentation. So I'm going to talk about microcystins in Lake Erie, what causes microcystins, and how can we forecast them. So let me get Zoom moving. There it goes. All right, so first, I want to talk about algae and microcystins. Just get everybody on the same page. Most algae are not bad for lakes. Algae are tiny plant-like organisms that are in the water column or living on the bottom of the lake. And half the Earth's oxygen is produced by algae. So every other breath you take, you should thank algae. Algae are the base of the food web. We have good algae that are food for small shrimp-like creatures that we call zooplankton. Get a pointer here. And then, so you have algae being consumed by zooplankton. Zooplankton are consumed by small fish and then bigger fish. There are hundreds or thousands of species of algae in lakes, rivers, and streams. They come in all different sizes, shapes, and color, but they all function somewhat similar. They need sunlight, they need fertilizers like phosphorus and nitrogen, and they need carbon dioxide. And they use these ingredients to produce sugars and oxygen. Lake Erie produces the most fish of all the Great Lakes combined because it has the most algae. Lake Erie has the highest nutrient concentrations, and it has the warmest water during summer. These two factors interact to support a really vibrant fishery like this walleye here. A general rule we say at Stone Lab and Ohio Sea Grant, it's not exact, but it's intuitive. Lake Erie has 50% of the fish, but only 2% of the water, whereas Lake Superior has 2% of the fish and 50% of the water. So Lake Erie and Superior are on opposite ends of the spectra when it comes to productivity in fisheries. And we thank algae for that. However, too much of the wrong type of algae is harmful. And we often hear the term harmful algal bloom. There's no consensus on what harmful algal bloom is, but the way I define it, breaking each word down, the word harmful means it has the potential to produce toxins. They may not always be producing toxins. They might be producing a lot or little toxins, but at least it has the potential to. There's harmful impacts on the ecosystem, and the toxins they create are harmful to human health, and local economies can suffer. So that's what the word harmful means to me. Algal is just describes what type of organism we're dealing with. In freshwaters, we deal with cyanobacteria, which are also called blue-green algae. Marine systems and the coastal oceans have harmful algal blooms that are very diverse, but an example of a marine harmful algal bloom is a red tide. And the bloom, again, not scientific term, it's just a general word that describes a biomass that exceeds what's normal. So a lake shouldn't be green, the oceans shouldn't be red. So harmful algal bloom is a large biomass of algae that has the potential to produce toxins and harm the environment and human health. I'm going to talk all about Lake Erie from here on out. Lake Erie algal blooms can be seen from space, from satellite. This is an August 2011 image. You see the western basin of Lake Erie covered with a type of cyanobacteria called Microcystis. We also see some harmful algal blooms in Lake St. Clair, mostly along the Canadian shoreline. But the western basin of Lake Erie can be covered in algae. The large central basin of Lake Erie off Cleveland and further east can often get the spillover, as you see in this image, of the western basin bloom spilling into the central basin. But for what I'm talking about today is focusing in on the western basin and the Maumee River, which drains a large agricultural watershed, empties into Lake Erie in the southwest corner. So a brief history of harmful algal blooms in Lake Erie. In the mid-1900s, Lake Erie was declared a dead lake by Time magazine. There was bad blooms, fish were dying because of low oxygen and chemicals, and the Cuyahoga River, which is in Cleveland, caught on fire several times. That prompted the US EPA to form the Clean Water Act and the Great Lakes Water Quality Agreement. All those targeted phosphorus bans and other types of water quality initiatives. By the 1980s, Lake Erie had recovered. The lake became the poster child for ecosystem recovery. Lake Erie became the walleye capital of the world, and there were no blooms. Since 2000, when we have we have really good data since 2002, there has been annual blooms documented. 2011, at that time, was the worst bloom in history, which is the background image of the slide. You can see a small boat going through a harmful algal bloom scum and leaving a wake behind. So 2014 was the Toledo water crisis, when the city of Toledo was told do not drink the tap water for 3 days because of high microcystin concentrations. 2015 became the new worst bloom in history. And in 2018, the Ohio EPA declared Lake Erie impaired under the Clean Water Act due to harmful algal blooms. And the word impaired has legal implications that should spur change within the watershed to help improve the bloom. Harmful algal blooms are just not a Lake Erie issue. They're truly a global problem. No matter where you are around the world, if you have a bloom, it's due to excess nutrient loading. And those excess nutrient loadings can come from fertilizers, they can come from manure, lawn care products, untreated sewage. Basically, if these things are put on land to grow plants such as crops or green grass, when they wash off the land and get into the water, they will grow algae. And if you have too much of those nutrients, you promote harmful algal blooms and cyanobacteria. Some are problems associated with those blooms. They produce toxins, which will be the main focus of my presentation. Taste and smell problems. So your tap water, even if there's not toxins in it, might smell a little funky. And if you're walking along the shoreline of a bloom, you'll get a smell of a strong sulfur compound. Negative economic impacts. My title slide, if you remember, was downtown Put-in Bay and a lot of lakefront restaurants and shops. When there's bad blooms, less people go to those restaurants and shops. We work closely with charter captains on Lake Erie, and they report about a third of their businesses lost during years with bad blooms. Cedar Point and other tourism around Lake Erie complains about blooms. The city of Toledo lost about $65 million during that 3-day do not drink advisory. The US, the property values in the US decreased by about $2.2 billion a year due to bad blooms. And there's food web implications. Cyanobacteria are not consumed by zooplankton, and the good types of algae die off. So we talk about toxins associated with algal blooms. This slide is from the Ohio EPA, and it lists relatively well-known toxins. Dioxin is probably one of the most toxic things that we know about. The ones in green are produced by cyanobacteria. So microcystin LR is more toxic than DDT, cyanide, and atrazine, for example. And when microcystins were first discovered, I don't know if that's the right word for them, but when they were first discovered, they called it fast death factor. They were discovered by injecting cells or killed cells of microcystis into mice, and they noted that the mice was dying fast, so they called it fast death factor. And then they later named the toxin microcystin. So microcystins are a group of toxins that are produced by cyanobacteria. They target the liver, and they target especially those with unhealthy livers. Microcystins can only be measured in water samples. We cannot measure microcystins from satellites. We cannot measure them with sensors, so we actually have to have a water sample in hand. There are over 200 congeners or different forms of microcystins, but their structure and function are somewhat similar. These different congeners, although very similar, have very different toxicity, which I'm not going to get into the toxicity part of my presentation today. But just know that microcystin LR, which we find in Lake Erie, is very toxic. But if you just make a minor adjustment and you call it and becomes microcystin RR, it has very low toxicity. And we can find both these toxins in the same water sample in Lake Erie. And there are several types of cyanobacteria that produce microcystins. In the Western Basin, we have Microcystis. When you hear about blooms in Lake Erie, everyone's talking about Microcystis. Planktothrix occurs in Sandusky Bay and large rivers and inland lakes. And we can have Anabina or Dolichospermum in Lake Erie, but it doesn't produce microcystins. Dulicospermum produces microcystins in other lakes, but not Lake Erie. So for all my work I'm talking about today, I'm talking about microcystis. So a general question we ask, is there a relationship between bloom biomass and microcystin concentration? So can you go out to a lake or a beach and see green water and assume it's toxic? Or the other way, if water looks relatively clear, can you assume that it's safe to swim in? So this graph shows data we've been collecting for the past several years in my lab. We have cyanobacterial biomass on the x-axis and total microcystin concentration on the y-axis. And overall, you see no general pattern, even though there is a significant correlation. But if you remove some of those outliers and focus where most of the data lies, you really see there is no relationship between bloom biomass and microcystin concentration. So this tells us if we're trying to develop a predictive model, we cannot rely on how green the water is. And also, cyanobacterial biomass doesn't equal microcystin concentration. But as I mentioned earlier, microcystin concentration doesn't necessarily equal toxicity. I'm not talking about the toxicity aspect of our work. I'm just going to focus on total microcystin concentrations because that's what is measured at the water treatment plants, and that's what the standards are based on. We assume if there's microcystins in the water, it's all equally toxic. So why is there no relationship between biomass and toxin concentration? Within a mixed community, within a microcystis community, there's toxic and non-toxic strains. We're in the news, we're hearing a lot about COVID strains and how different COVID strains have different impacts. The same is true for cyanobacteria. There's lots of different strains of cyanobacteria and bacteria in general. But out in Lake Erie and many other lakes, it's a mixed community of toxic and non-toxic strains. In order to be a toxic strain, you have to have the genes needed to produce microcystin. So these 5 cartoon cells I have here, 3 of them have the toxin gene that I have indicated by this little piece of DNA, where these 2 other cells are non-toxic. But also the amount of toxin produced by a toxic cell can vary. So this toxic cell is making a lot of microcystins. This one made a few, and this one only made 1. So understanding how the microcystin to biomass ratio changes can be helpful. There's a lot on this slide. I'm going to take some time to break it down. So this slide I'm pointing out here on the right, the red triangles are the ratio of microcystins to cyanobacteria biomass. We see early on there's lots of variability, but overall there's a seasonal decline. We have high ratios in July and August, low ratios in September. And this follows nitrate concentration. Nitrate is the most common, the most abundant form of nitrogen in the water. Every year we see a seasonal decline in nitrate concentration, and that corresponds to a seasonal decline in the toxin to biomass ratio. The graph down the bottom looks at the toxic to total microcystin ratio. So in July, early August, between 80 to 90% of the cells in Lake Erie have the toxin genes. As nitrate gets depleted from the system, the amount of toxic strains also decreases to less than 10%. So throughout the summer, we see more toxic strains early summer, less toxic strains in the fall. And we see this every year in Lake Erie. We can also do experiments where we take a bloom sample and add in nitrogen or phosphorus and see how microcystins respond. When we do these experiments, we see that when we give them nitrogen, if it's nitrate, ammonia, or urea, and they need high light, we get a the biggest jump in microcystin concentrations. When we only give them phosphorus, they don't change from the control. So in order to produce microcystins, they need abundant nitrogen and light. Nitrogen only won't trigger toxin production. They need nitrogen and light. Cyanobacteria biomass can be measured from satellites at large spatial scales with high resolution. Note this is biomass only, okay? Each image here shows a snapshot of the largest bloom extent in Lake Erie. So 2002, there wasn't much of a bloom at all. 2003 was a large bloom. This 2003 bloom got the attention of scientists in the area. And then we see some year-to-year differences. 2011 was a fairly large bloom, and this bloom spread out to the central basin off Cleveland. 2012 was a small bloom year. 2015 was the worst bloom year, and we see some year-to-year differences. So if we take every image throughout the summer and add up all the pixels, we can create a bar chart. Oh, hang on, I forgot. Before I get to that, I'm going to say we're going to look at, can we make similar maps for microcystins? For that, I apologize. I did a presentation yesterday where I used slightly different slide order. So I'm going to ask, can we make similar maps for microcystins compared to the biomass maps? So what we did in 2018 and 2019, we did an intensive one-day survey of the Western Basin. Several universities, agencies, local, state, and federal agencies helped out, US, Canadian, where we all sampled a different area of the lake. We did this on August 9th, 2019 and August 8th, 2018. The color image on the back is a biomass image from satellite. And a smaller bloom in 2018, and we had a smaller number of groups involved. So this is what we called the HABS grab, where we all went out on one day and collected as many samples as we could. Some images from this sampling event, I called it grab and grow sampling. There was no onboard filtering or anchoring. Once the boat got to the station, we grabbed a water sample, measured surface temperature, and moved on to the next site because we wanted to get these samples in back to the lab as soon as possible. In 2018, we collected 100 samples. In 2019, we collected 172 samples. All the samples on the US side were taken to the University of Toledo Lake Erie Center, where they were processed, filtered, and aliquots poured for different parameters. And the Canadian samples went to the University of Windsor. So this is the maps of microcystins for 2018 and the event on 2019. We see some differences right away. 2019 had much higher concentrations than 2018. And the location differs too. The highest concentrations in Maumee Bay and along the Ohio and Michigan shoreline, whereas 2018, the highest concentrations were along the Ohio shoreline and out towards the Bass Islands region. 2019, there was very little microcystins out by the Bass Islands. So this is the maps of microcystins for those 2 days. If we look at 2019 closer, we see very high gradients over short distances. At the bloom boundary, we go from concentrations about 10 parts per billion down to below detection. And in Maumee Bay, the highest sample was 46 parts per billion, and just a couple kilometers away, we're down in the 2 to 5 range. So these rapid gradients in the bloom boundary are very hard for models to capture. And also, if you look at the microcystin to chlorophyll ratio, which I talked about earlier, I showed how it changes from high ratio to low ratio throughout the summer. if you look at it spatially, we see just as much variability. Along the Ohio shoreline in 2019 had the highest ratio, so the cells here were producing more toxins than cells further out in the basin, but a different pattern in 2018. Along near Oak Harbor and up by the Bass Islands had the highest toxins to biomass ratio. And if we plot that ratio versus nitrogen concentration, we see a significant correlation. At higher nitrogen concentrations, we had higher toxins per chlorophyll. So again, nitrogen is important for microcystin concentrations. So that was great. We made those two maps for those 2 days where everyone was out on the lake on one day. And we wanted to ask, can we use historical data to map microcystins in previous blooms? Fortunately, there's lots of data in Lake Erie. Water treatment plants are collecting data at least weekly, sometimes daily. There's many research groups, NOAA, University of Toledo, USGS, Ohio EPA, OSU Stone Lab going out weekly to monthly. And we also work with charter boat captains who collect samples for us weekly. So there's lots of data available for Lake Erie. And we asked, can we use that historical data to make maps of microcystins in previous blooms? However, once we got in the data set, we quickly realized there's challenges, and mostly associated with spatial interpolation when few samples were collected. August 1st, 2016, there were 16 samples collected. But these, between this sample near West Sister Island in the middle of the basin and out towards the Bass Islands, that is a huge gap. And when we during the HABS grab, we saw that gradients can change rapidly over short distances. We cannot extrapolate between points very well. And it gets impossible when you only have 2 sample points collected in one day. So what we did to try to overcome that is we took all data from 1 week and compressed it to 1 day. So all the samples that were collected in the week, we artificially said they were collected on Monday. And then we used the satellite data to map out the bloom edge and said everything outside of that bloom edge is 0. So August 14th, 2017, here's the map of microcystins. However, when we compare it to the bloom biomass data, we see there's not much agreement, right? There's high biomasses along the Michigan shoreline, but yet we're not seeing any microcystins there. We've seen bloom biomass kind of go up in this arm reaching up towards Ontario. We don't see that reflected in microcystins. August 27th, the same year, a little bit better agreement, but we're missing some of these outreaches of the blooms, so they're not lining up. However, when we look at the map of microcystins on the HABS grab and compared it to the bloom biomass from satellite, we do see that they do match, they do align. So when we have a high number of samples, the spatial patterns are much better. So what this tells us is that the routine monitoring in Lake Erie does not have the spatial resolution needed to inform models on a basin-wide scale. So this HABS grab data date had 172 data points. Routine monitoring, we might have 20 samples, and we're missing large parts of the lake. And what's happening is there's simply just no samples that were collected in this area, in these 2 areas. So we can't extrapolate what we know to where we don't know. It's impossible to do. So challenges to forecasting microcystins: low amounts of data to initialize models, and then we have very low once we do run models, it's hard to compare a model output to observed data because you're dealing with low amounts of data. The bloom boundary and the rapid concentration gradients within the bloom are difficult for models to capture. And there's no relationship between microcystin concentration and biomass. And that ratio between the two is not spatially consistent or temporally consistent. So how do we build a microcystin forecast given all these challenges? We know that we can forecast bloom biomass on a seasonal scale. And when we go back to that satellite image that I mentioned earlier, where we had all the years, and I said if you add up every image from every pixel, you can create a bar graph. So here's that bar graph that was referenced earlier. So 2002 was a small bloom. 2003, I said, was a bloom that got everybody's attention. 2011, 2015 were really bad blooms. However, when you see this year-to-year difference, but this year-to-year difference is based on the amount of phosphorus that comes down the Maumee River during the springtime. In 2011, 2015, they were very wet years, a lot of phosphorus load and large blooms. So if we know how much phosphorus enters the lake during the springtime, we can know how much bloom biomass is going to be in the lake. However, when we try to do the same thing for microcystins, there is no relationship between phosphorus load from the river or nitrogen load from the river. Researchers at NOAA use chlorophyll concentrations and ask what is the probability of microcystins exceeding a certain concentration. They used satellite data, and then they used weekly weekly samples that were collected by NOAA researchers. They looked at the microcystin to biomass ratio within water samples, and then applied that microcystin biomass ratio to the satellite data to make a map of microcystins. So here's their map of microcystins. However, their main caveat is they don't have data all throughout the basin. NOAA samples mostly just the corner of the basin. So they're extrapolating well beyond their observed range. And then for a given chlorophyll concentration, they said, what is the probability of microcystins exceeding, for example, 0.3? Our idea was to take maps of microcystins, for example here, and plug these concentrations into water movement models. So this is what they call the FVCOM model. But all the little arrows you see here are direction of water flow in Lake Erie. Water enters in through the Detroit River and the Maumee River, and in general, it moves west to east. But there's some differences in patterns. The wind can affect that. If we get a strong east wind, these arrows will be pointing more towards the east. If we have a strong west wind, they'll be pointing more towards the west. So water moves around on Lake Erie, and we assume this water moving would carry microcystins. So if we know where microcystins are today, and we know where the water is moving, we should be able to issue a microcystin concentration forecast several days into the future. And then what we thought we could do is compared our model concentrations to the observed concentrations 1 week out. However, as I mentioned a couple minutes ago, that the lack of data to start the model and then the lack of data to compare the models was a strong, large hurdle for us to try to overcome. So knowing that, we confined our efforts to a smaller area that has more data per area. We also wanted to look at, can we incorporate microcystin production into the models? So the previous slide, if you just know concentration and where the water is going, that doesn't account for biology. We asked, if you include microcystin production rate into the models, can you improve the model? So in order to do that, we first had to quantify production rates. Judy Westrick at Wayne State did all the microcystin analysis here. We did this experimentally. We had an ambient nutrient control, and then we spiked in phosphorus and nitrate, phosphorus and ammonia, phosphorus and urea, and measured microcystin concentration over time. And then basically the slope of that line is the production rate. So if we take that production rate and plug it into the models, will our models be better? And what we saw is when the models with microcystin production rates performed better than models without microcystin production rates. These graphs or these charts are quite complicated, but I'll step you through it. On the Y column, these are NOAA GLORAL sampling sites. So 8 sites in the Western Basin. WE12 is at the Toledo water intake. The top graph is with microcystin production. The bottom graph is without microcystin production rates. And then on the X, the columns are just dates the models were began. Okay, so there's two, well, when you run a model, there's 4 outcomes. The model, and for this question, we asked, did microcystin increase or decrease over 1 week? So there's 4 outcomes. The model indicated there would be an increase in concentration, and the observed data indicated an increase in concentration at the site. So dark blue is correct. On the other hand, you can have the model indicated a decrease and the observed data also decreased. So that is a light blue model, a light blue, also correct. So in these figures, blue and dark blue is correct. Dark gray is a false alarm, where the models indicated an increase in concentrations, but the observed decrease, and then a missed event is light gray when the observed went up, but the model went down. So we see with microcystin production, we have more dark blues. And specifically, if you look at WE12, the Toledo water plant, our models with production rate was 88% accurate, just simply asking, will microcystin increase or decrease over the next 7 days? And that information alone can be very helpful for a water plant operator to better prepare, to get ready to better prepare and better treat microcystins in the tap water. We also see there's some differences among the years. We did much better in 2019 than the model did in 2018. 2019 was a very large bloom, and it's easier to forecast a large bloom than it is a small bloom. Kind of like the snowstorm we're dealing with right now. When you have a giant snowstorm heading your way, it's likely that you're going to get snow. Whereas if it's a really small snowstorm, it might miss you, right? So large blooms are much easier to forecast than small blooms. We also got similar results when we asked, would microcystins exceed 1 part per billion or 5 parts per billion? Okay, I already said this. So here's some suggestions that I have to improve microcystin capabilities. With the absence of microcystin data between 2 sampling locations, using the microcystin to biomass ratio that's observed of nearby locations, and then the satellite data is likely the best current method to estimate microcystins at an unmonitored location. And currently, I think forecasting the probability of microcystins exceeding a certain concentrations or asking will microcystin increase or decrease over the next several days is most feasible. The best we can do right now is 1 week out, because that's how the available data works. We had to compress it all in 1 week, so we can't issue a forecast yet of 4 days out because we don't have that good of temporal data. But we do believe if we had better data, the forecast would be much more accurate. And we need more microcystin data, especially further away from Maumee Bay. Maumee Bay is near Toledo, where the Toledo water intake is. Repeating the HABS-GRAB surveys would be really beneficial, especially early peak and late bloom stages. We also need to know more. About what controls microcystin production rates. We saw with 2 sites and coarse temporal resolution of our experiments, plugging those into the model gave initial promising results. So if you have a better understanding of production, we can plug that into a model and get better results. We also need to look at the role of light or water clarity on bloom toxicity. It's more than just a phosphorus and nitrogen issue for microcystins. We know toxic strains have higher light requirements, and we see in experiments that Microcystis produces more microcystins at higher light. And a real world example, that 2015 bloom that had massive biomass, it had relatively low microcystins. The lake had high concentrations of phosphorus and nitrogen, but the lake was also very turbid. It was very muddy. We had a lot of storms in 2015, high wind storms that mixed up the bottom of the lake. And throughout the entire summer, non-toxic strains dominated. So there is an impact of light. We need to better understand it. So with that, I'd like to thank the main funding source for this project is the NOAA EcoHab program. My colleagues at Limnotech and Bowling Green State University, University of Toledo, and University of Michigan, and Judy at Wayne State helping us out. We also received funding from Oceans and Human Health, the NSF and NIH award that's at BGSU. Several state agencies have helped fund this research. And that HABS-GRAB data set that I mentioned has been published in the journal Harmful Algae. So with that, I'll be happy to take any questions that you may have. Thank you.
Thank you so much, Justin. That was a really great presentation. For those of you who actually attended our seminars last semester, Dr. Judy Westrick actually spoke a little bit on this project, on one of the projects, the project that you were talking about, the 2018-2019 sampling. Her lab actually did the testing of the samples. And so, yeah, I'll post a link to that in the email that I send after this with the link to this presentation. But yeah, when I had initially asked you to present, I didn't make the connection. It was, as I was like looking at the maps, I was like, wait a second, I actually recognize these maps. So yeah, I'll post the link for anyone who's interested in that. But thank you so much for speaking, Justin. You said this was a really great presentation. If anyone has any questions, feel free to throw them in the chat. We already have a couple questions that have come through. So one is, are the toxins worse in the late summer when there are less nutrients compared to the early summer when the nutrients are higher?
That's a really good question. So I didn't talk about toxicity, the actual how toxic the microcystins are in the different congeners. There is also a role of nitrogen there too. So early summer, we might have high concentrations of toxins, but it's going to be a mix of RR and LR. So the overall toxicity of a water sample is, it depends on the congeners. But if you have a mix of a low toxicity and a high toxicity congener, you end up with, let's just say, medium toxicity. Late summer, we have more microcystin LA and still some LR, but no RR. LA and LR are just as toxic as each other. So you might have lower toxin concentrations, but you might have the same toxicity because the toxicity of the toxins is higher.
Okay, cool. Thanks for the clarification. We have a couple more. To what extent would your modeling be improved by having pure authentic standards of different MC congeners?
Right. So the modeling work is based on the ELISA method, which I didn't talk about. The water plants in Ohio and the Ohio EPA based all their standards on the ELISA method. The ELISA method doesn't tell you if you have LR or RR or LA present. So you just assume that it's all toxic, and you assume the standards in the ELISA kit are microcystin LR. So if you're getting hits on microcystin on the ELISA, you're supposed to assume it's toxic. Judy Westrick's lab does LC mass spec, which has, I think she has 13 or 14 different congeners. So that gives you the information on how it gives you the concentration, but also the congeners, so you kind of get an idea of toxicity. For the modeling work, we went with the ELISA data because that's what the water plants deal with. They deal with the ELISA data, and that's what all the historical data is based on, is ELISA. So that's how we're focusing the modeling work. But there is a caveat that toxicity doesn't equal toxin concentration. So having more standards would help, but we need more of that data.
Yeah. Okay. Do you have any plans to do another large scale sampling event?
We would like to, but they're kind of pricey in funding. That was the main issue is funding. It's extremely challenging to get everyone out on the lake on one day. Mm-hmm. To do this, we said, but in the spring we picked two weeks. We said we're going to target the first and second week of August, based on bloom conditions and weather forecasts. That's how we picked a date, and we picked a date basically 48 hours ahead of time. And everyone during their meetings agreed upon, if you pick a date that you can't go out on, you're just not going to get sample. You're not going to collect samples that day. We're not, if 9 out of 10 people can go out, we're going to go out when 9 of the 10 people can go out. So that was challenging. It's harder to do it late August and September when school gets back in. The grad students and professors are less available. Mm-hmm. But we would like to repeat it. It's just we need to get the funding for it.
This actually isn't a question that came into the chat, but it was something I was thinking about when you were presenting. So you went out you went on one day to collect all the samples, and then during the other, when you're looking at the historical data, it was more like, or during one of the studies you were talking about, it was, maybe like throughout 1 week. Mm-hmm. So with the harmful algal blooms or the microcystin concentration, would that change a lot, like day to day? Would you see, potentially a big jump from one day to the next, depending on, the cloud coverage or, something else that could be a factor?
Yeah, absolutely. You can, especially on the bloom boundary. Okay. If you just draw a imaginary straight line, say the bloom boundary is, if you draw a straight line down the middle of the basin, and that's where it is today. If you have an east wind, that boundary is going to move, and it's going to move closer towards Toledo. if you have a strong west wind, that boundary is going to move further east towards the islands and further east.
Okay.
So that's another challenge. The modeling that we did, we took all data available to make our maps, but we only looked at the NOAA sites, which are in the western corner.
Mhm.
So hopefully, that bloom boundary layer is beyond the NOAA sampling range, but yet we have limited data. We have some data, but limited. So the bloom boundary layer hopefully is not swinging back and forth across that. But the current models are ran for 7 days, and those current models would pick up the bloom boundary moving.
Okay.
It's just that what is more concerning is if you have calm weather, calm waters, I guess, the currents aren't moving very fast. The bloom's just growing. So if it's here on day 7 and the water currents aren't moving it, you can have expansion by growth that you may not pick up during 1 week per sample or 1 sample per week. So that's a little bit more challenging because you can't pick that up on the water currents alone moving things around.
Okay, cool. Okay, here's another question. Questions around evolution. Were cyanobacteria that produce toxin more prevalent in distant past? What is the purpose, the purpose of the toxin? And
was there a follow-up?
Go on.
Okay,
yeah, there's one follow-up, but you can go.
All right, yes, that's a really good question. I get that question a lot. The MCY operon has been mapped or dated before metazoans are around. So cyanobacteria were producing microcystins before things were trying to eat them. So that rules out the hypothesis that microcystins are defense against grazing. So they were producing microcystins well before. The current and probably most agreed upon hypothesis is that microcystin aids in oxidative stress. So when microcystis is at the surface of a water column, it's getting blasted by light, and they can be producing reactive oxygens, and microcystins bind to proteins to help stabilize that. So early summer, when there's more light, the sun's higher up in the sky, photoperiods are longer, water's clear, there's a potential for more oxidative stress early summer. So the toxic strains do better, they're selected for. Whereas in September and October, the water's muddy, there's less light, shorter days, less risk of oxidative stress, and then the non-toxic strains then outcompete the toxic strains. So I hope that answered the question.
So the follow-up question to that is, does the toxin produce selective pressure on zooplankton, more resistant?
The effect of microcystins on zooplankton is somewhat mixed. The reason why it's not clear is that if you take a toxic strain and a non-toxic strain, or if you take a toxic strain and knock out the microcystin gene, they all have the same impact. They all have the same negative impact. So there's other, cyanobacteria produce a lot of things. They're just not producing microcystins. They're producing things that disrupt endocrine systems. I'm familiar with a study of new or still might be ongoing, but they're seeing that zooplankton are producing more eggs when they're exposed to microcystis, not necessarily toxins. They're producing more eggs, but then the offsprings are producing less eggs. So there's some sort of a carryover where right now you're producing more offspring, but your offsprings are less fertile, I guess.
Yeah.
And that's not tied to microcystins. It's tied to something else that we might not know what's happening yet.
Okay. Okay. Have the toxicity and general growth data been correlated to actual weather patterns? So this was kind of what I was asking before, like sunny days, for example.
Well, they need, when we looked at the growth rates and the production rates over time throughout the summer, we see that the highest growth rates and the highest production rates occurred early summer. Early summer, there's more nutrients and the water's clear. Late summer, we got lower production rates. And when we added nitrogen back into it, we didn't reach the same production rate that we did early summer. So that tells us that it's not just nutrients, it's also light issue potentially, or the community structure is different. So we had more non-toxic strains late summer. So it's a mix of nutrients and light. Temperature can also be an issue, but different studies are giving different results. Some show that cool temperatures favor toxin production. Others say warm temperatures favor toxin production. But the issue is looking at individual strains, individuals toxic. There's not just one toxic strain. There's bunches of them, and they might be behaving differently in response to temperature. So that's a tricky topic, but they all pretty much respond to nitrogen and light about the same.
Okay. Let's see. I think you actually did touch on this, but you can maybe repeat it. Is nitrogen the main nutrient driver of harmful echo blooms? How about phosphorus?
That's a good question. We know that phosphorus is critically important. If you don't have the phosphorus, you're not going to have the blooms. So the Great Lake Water Quality Agreement, state of Ohio, state of Michigan, Ontario have all agreed to a 40% phosphorus load reduction to minimize blooms. If we do that, we will dramatically lower the amount of blooms, but it's still a question on will it be, what impact will they have on toxins? If phosphorus goes down, but nitrogen stays the same, if you have lower biomass in the summer, lower biomass into late summer, the water is going to be clear. And if there's lower biomass, there's going to be more nitrogen persisting into late summer. So you might be selecting for more toxic strains. But then the question is, would the difference in biomass offset the higher presence of toxin strains? So yeah, we'll make the water less green, but it might actually make it a little bit more toxic per cell.
Okay.
But at what levels would toxin production late summer be ramped up to an extent that it cancels out the phosphorus load reduction? In general, in limnology and lake management, there's a debate upon, do we manage phosphorus only, or do we manage phosphorus and nitrogen? It's a large debate, but managing phosphorus is first and foremost, and then would nitrogen increase the bang for the buck?
Okay, so it's getting close to 3:30. There are a lot of questions still. People are very, very interested, which is great. Let's plan to go through a few more questions. We'll go a little bit over.
That's fine.
I'll try to kind of summarize some of them, try to combine some of them if I can. So this one question, has anyone done research viewing spectral shapes through decomposition of the satellite imagery and seeing main components? This person's saying this would show specific components such as sediments and things like phycocyanin and other pigments, then correlating those to microcystin production. Does that make sense?
Yes, that doesn't. All that's been done. The satellite images I've shown are based on chlorophyll, but they're based on chlorophyll when phycocyanin is present. So you can look, you can see spring diatom blooms from satellite. But if there's no phycocyanin present, we can determine that's not cyanobacteria bloom. So the biomass images that you saw are based on chlorophyll when phycocyanin is present. Because once phycocyanin is present to a certain level and you have high chlorophyll, it's going to be a cyanobacteria bloom. You don't have a big diatom bloom with some cyanobacteria mixed in. It's the other way around. You have a big cyanobacteria bloom, and there may not be other things mixed in. As regarding for microcystins, we can't measure microcystins from satellite. Most microcystins are tied up within a cell, so they can't see inside a cell. But we should use the satellite images to see where the bloom is, the bloom boundary layer is. And then if we have some idea of where the most toxic cells are, most toxins per chlorophyll from a water sample, then you can get an idea of how toxic that individual pixel is and then extrapolate from there.
Okay. Okay. Does the biomass move up and down the water column? And does this change the toxin concentration with that movement? I'm assuming when they say, does it change the toxin concentration, they mean at the surface?
Right. Good question. Is my PowerPoint still up?
It is, yep.
Yeah, so you see on a calm day, microcystis will accumulate at the surface and form a surface scum. If you go back, if you're not against the shoreline, just imagine this is what it looks like offshore. You see the scum. If the wind comes by, it mixes it all up. That biomass is still there. It's just more equally distributed, and the toxins will follow that. They'll be equally distributed throughout the water column. The models that we use account for that. So they're a 3D model. So they account for where microcystins are in the water column. They account for where the microcystin was actually collected from. Unfortunately, there's no standard method for sampling, for collecting a water sample out in the lake. My preferred method, and what the University of Toledo does, is use an integrated sample that goes from the surface to bottom. So you take a whole slice of the water column, and with that method, the water sample you get, it doesn't matter where the bloom is, if it's at the surface or mixed, you'll get the same data. But if you start using individual points throughout the water column, you may miss a bloom. If you have a thick scum like this, but you collect a sample only halfway down, you lower a little contraption to grab water at a certain depth, you won't collect this. You'll have a much lower number. So fortunately, models can account for that. The current models are really good for Lake Erie, which is really fortunate for us.
Yeah. Okay. Let's see. Can high sunlight affect photosynthesis P2, thus stress the organism and result in stress and release of toxins?
Right. Algae in general are lower light adapted than plants on land. Trees and grass are exposed to full sunlight and they do fine. Microcystis doesn't like to be at the surface. And if they stay there too long, if it gets too calm, they can actually get trapped. Their photosystem too gets damaged. Then they can't photosynthesize and they can't produce sugars. And the sugars allow them to sink in the water column. So a healthy cyanobacteria cell will come up to the sunlight, photosynthesize, accumulate sugars, which changes osmotic pressure, and then the cells sink deeper down the water where it's dark. They respire the sugars and float back to the surface. So a healthy cell will go up and down throughout the water column. If it's too calm, they can get stuck. They photosynthesize, and if they die in lice, then toxins can get released to the water column that way.
Okay. Okay. I think we got most of the questions. There were a couple, but I'm gonna wrap it up. Thank you so much again for speaking. I really appreciate it. This was a really interesting talk, and like I said previously, Dr. Judy Westrick's talk is kind of connected to this, so I'm going to add it on to the email that I send out with the link to this event. I do want to say that, so thank you again, Dr. Justin, I appreciate you talking. I'm gonna just say like you said, a recording of the event will be sent out following the event. And then our next Water at Wayne seminar is going to be on February 10th. And Dr. Manuel Elias Gutierrez, he will be speaking on DNA barcoding and methods to uncover freshwater biodiversity. Some examples from the neotropics. Everyone that's already signed up for the seminar they'll get the invite. You don't have to sign up again, but I will send out an invite for that, more information about that regardless. So thanks again, everyone, and enjoy the rest of your week. Thanks. Bye.
Thanks again.